Haley. How's it going?
Oh, so good. I'm so excited about this topic because I feel like we've touched upon it. It's kind of come up in other conversations we've had. We both deal with something called the CSI effect in our work in very different ways. We've got very different interactions with, how this impacts our work and people's perception of our work. So I'm excited to dive into this with you. I first would think it makes the most sense to define what we're talking about with the CSI effect. I don't know if there's like an Oxford Dictionary definition of it, but if you want to walk us through what the CSI effect means in your lens.
Yeah. I think before television shows that popularized crime scene investigation, which is, CSI, think dealing with crime scenes and physical evidence and bodies and all that kind of stuff was seen as a little bit creepy. I think they in movies and things they would show you see like the guy that works down in the morgue and he was always like super creepy and just to kind of add to the effect. But then movie or TV shows like CSI and things like that came out and they really started to glamorize what these folks do. And so we sort of referred to that whole thing as the CSI effect. I remember back when I was at University of North Texas, they had the local museum had a uh, a CSI exhibit. It was like this traveling exhibit that went all around the country, and it would have, staged crime scenes, and the we would walk sixth grade students through there and explain all this stuff. And we donated um, shells of equipment so that you could see what the real thing looked like. The CSI experience, I think was what it was called. And so it started to be cool.
Mm.
And it started to be more known some of the things that go on in a crime lab or when people are working at a crime scene. Um, this had a lot of positive impacts that I saw because instead of it being perceived as a little bit creepy to do these kinds of things, it started to be cool. And so we had a lot more students to choose from because they were interested. They wanted to come do a forensic genetics master's degree or something like that. And so it really increased the available student pool because it started to be kind of cool instead of kind of just creepy. Um, but in terms of so that was a positive effect. More folks, bigger student pool. You get more people, more bright minds that want to come do this stuff. I think also it familiarized sort of the general public with DNA evidence, um, and what it, or at least what the television show told you it could do. And I think that's where like there were pros and cons.
Yeah.
A lot of it was positive. Um,
Yeah,
I
because
don't
with
know.
a
I
with a
mean,
spotlight, a more direct spotlight on this work also opens up more room for opinions and thoughts on that
yes.
work and how things should work because that's how it worked on the TV show that I watched last night, kind of thing. And that's really the CSI effect that I think probably impacts your day-to-day work or the scientists in labs today working on criminal investigations is, well, I saw it happen in 30 minutes, but of course we know it doesn't happen like that
Right.
for real.
Yeah. And I think that the misconceptions we would see also with new students coming in because they thought they were gonna interrogate suspects and chase suspects and work the crime scene and work the evidence in the lab and testify and that's like six different people's jobs most of the time.
Mm.
So yeah, the it had a lot of positives and I'm glad that it shed light on the very hard work that a lot of people do. But just like anything else on TV, you got to realize it's not 100% real. I'm not going to watch a pie bake for 45 minutes on a cooking show, nor am I going to be mad at Martha Stewart for not making me watch it. Pie goes in, pie comes out. It's done.
Right.
Amazing. So I think, that's uh, a lot of the kind of differences between reality and CSI, other than, you know, it's not just one person doing all those jobs, but the time it takes is very different.
Are there other misconceptions that have revealed themselves to you through like people's questions about your work? Are there any questions that you get that you're constantly answering to kind of demystify what you really do or what you really did?
Yeah, I think so. I think people hear DNA and they think it's all one thing.
Mm.
And it kind of is. If you take one of my cells, it's got a nucleus that has a bunch of DNA in it, and it's got a mitochondria, or it's got a bunch of mitochondria, and they all have their own little piece of DNA. If you take all that and extract it, then you have my DNA, right? But what can you tell from that? Well, it depends. It depends on what you're looking at. So if we're talking the FBI's National DNA Index System, which is the database that's run by the software CODIS, where we put offender profiles and crime scene profiles in to see if we get a hit, we're only looking at 24 things. And those 24 things were specifically chosen not to give you any other information about that person other than to tell them apart from another person. So when you see a TV show, they're like, oh, we have his DNA. And now we know he's 5'10 with, one green eye and one blue eye and sandy brown hair that he likes to part on the side. And, and he's a Pisces. And here's his driver's license picture and his fingerprints and all. No, you don't get all that. I think that is part of, that, oh, well, they have the DNA, and that should tell you everything, but it really depends on what you're looking at. And so the gold standard for telling people apart really only looks at 24 things, and it is really only good for telling people apart and for telling very close relatives.
Mm.
And so anything beyond that, you're going to have to use a different technology to look at more things in a person's DNA to tell more about them. I would say that's like one of the main, because they're like, well, they have the DNA profile. Why don't they see if they can find a match in a genealogy database? You find his relatives, and you're like, well, that DNA profile that tells people apart is not good. Is not the same kind of thing that goes into a genealogy database to try and find more distant relatives.
Hmm.
You have to use a different technology, and so DNA is not just DNA is not just DNA. It depends on how many things you're looking at and which things and what you're doing with them. I think that's probably the biggest misconception.
Right. Yeah, and admittedly, I don't watch a ton of actually I don't watch any these days of dramatized crime dramas,
I
TV shows.
can't.
And partly because I spend so much time in the reality of this in my storytelling. I have a true crime podcast. I'm an investigative journalist. I've seen it for real, and I don't want to watch the dramas. And what I've found where the drama, when I have watched the TV dramas, what they do skip over is that even if there's a match, a hit, that that doesn't close the case. And you don't go from match to arrest. And one of the things I've been trying to, walk people through when I'm dealing with a case that's, has had an arrest or is about to go to trial, or if it's a solved case that I'm covering for whatever reason, is explaining that that's one piece of the puzzle, this match, this not being able to exclude someone from the suspect pool. But then there's a lot of other, boots on the ground investigating that goes with it. There's, hopefully a cheek swab that can confirm it, particularly in the case of genetic genealogy. If you've identified someone in their lineage, well, you need another sample to confirm that that person is in fact a direct match. And then it's looking at the circumstances and witnesses. You can't just go to trial with the DNA sample. And that's where the TV dramas they've cut out that chunk of like less sexy investigating, because that part before the commercial break, when it's a match or it's a hit, and then after all the commercials you see who they're talking about, that suspect in cuffs or whatever. That's been my experience. What I've been trying to, podcasts are in this unique position to really educate listeners and roll it back, because it is true crime, not dramatized crime. So I've been trying to do that, but it's also been a learning experience for me, having grown up in the time of these crime dramas that my mom always had on the TV that I would run through the living room because I didn't want to see it because it freaked me out. So interesting that this has become my career, but that's the part that I've identified, that misconception where a match equals arrest. That's not the case.
Nope, not always. Yeah, there's a lot of parts missing. I have trouble watching those because they'll take an object and just like put it in a microwave. Then, oh, there's the profile. We're done. And the processes they don't always have the right equipment, and that's fine. Like, they're doing the best they can. But yeah, just sort of inaccuracies in how you actually get from, even just how you get, not even all the way to an arrest, but how you get from an object with some kind of biological material on it to a DNA profile of any kind.
Mhm.
Watching that go completely sideways, or they'll have stuff that's like amazing cool, there's like a 3D rendering of the whole person floating there, and you're like, well, that would be neat, but it's not how not how it works in my world anyway.
Uh,
um
I'm
yeah.
curious, you've mentioned this in passing in previous conversations offline, that you had an experience as a consultant. I don't know how much you can talk about that, but did you worked on a set, or tell me about what that experience was like?
Oh, yeah, a little bit. So at the time, I was working for a company that made DNA sequencers for crime labs, and TV show wanted one of the sequencers for the show. And so I think it ended up coming to me because nobody else wanted to bother talking to them. I don't know. So I think I kind of got voluntold. And so I did. I found him a non-functional sequencer box, at least the outside part. And I got to talking to him, and they were like, "Oh, well, we want to do here's kind of parts of the storyline, and we want it to do, this is gonna be like super cool futuristic technology. We want it to do this, this, and this." And I'm like, well, items 1, 2, and 3, we already do. If you've got a medium-sized sequencer, you could already do hair color, eye color, um, and basic biogeographic origin kind of ancestry stuff. I was like, yeah, no, that's already happening. And then
Did that
they were
blow their
like,
minds,
oh,
or?
okay, so can you talk to us more? And I said, sure. And yeah, I ended up going to the set. It was just way before the actors show up, and it was in the middle of COVID. So they're like, COVID test you 14 times and then you get to go, and then they kick you right out. But I was trying to help them set up the lab, and I'm like, okay. And a bunch of other companies had given them stuff to be on set because, of course, I mean, that's cool. You want your robot to be on the TV. That's cool. And I was like, okay, first of all, this thing, and this thing, I'll do the exact same thing. They're just different sizes. Also, like, labs are generally divided into pre-PCR. So PCR is the polymerase chain reaction, which is a way to make copies of DNA. So there'll be pre-PCR where you handle all the evidence and do the DNA extraction. And then there's post-PCR. You want that to be completely different because once you make a billion copies of DNA, it would be really easy to contaminate back to a piece of evidence that only has a few copies of DNA. So you want to keep those things forever separate. And I was like, yeah, not only would this not be in the same, like, not only would this not be on the same bench, but it's not in the same room. You gotta move these things. At least make it look separate. Um, the person that they had hired as their science consultant that they were paying, which in retrospect, maybe I should have asked to be paid, but I didn't. Um, was a very nice person, but they were a crime scene tech and they had retired, I think 8 or 9 years ago, which in the DNA world is a lifetime. That's a long, yeah, I mean, lots happened.
Right.
And so they would be telling, oh, well, at this point, they would, run a gel. And you're like, oh, we haven't done that in so long that most labs don't even have a thing to do with, you know. Um, and so, and it's I have no idea how they find the people that do consult, like for money.
Mhm.
Um, I know how I got found. It was just because I happened to be working for the company that they wanted a thing. But yeah, I helped them set up the lab. And then I talked them through the scripts. They would call and they would say, okay, so they're going to do this. Is this right? Are they going to say that? And is that right? And I would talk them through it and be like, okay, yeah, this looks good. This part, no, that's not how that works. And this is how you would say it, or this would be the correct jargon. I think the, the worst. I was all excited. I'd flown to California, had gone on set, helped them set up, talked them through the scripts as much as I could, sent them graphics of what the software would normally look like, that kind of thing. And the first episode was coming out. And so I told everybody at my company's headquarters about it. And I'm just like DNA, just like anything else, we use a bunch of jargon and we have acronyms and things that we use. And I don't know why, but when we talk about short tandem repeats, which are the DNA profiles that go in the FBI's ENDIS system, we call them STRs. You say the letters. If you're talking about a single nucleotide polymorphism, which are really the things that we look at for like genetic genealogy profiles, we make it a word and pronounce it as SNP. It's like, it just is. I didn't make it up. It wasn't me. That's just sort of how it ended up. So first episode comes out. I'm all excited. I got everybody at headquarters watching it. And the first thing they say, like one of the first things when they're in the lab, the actress lady did such a good job. And she's like, well, normally we wouldn't be able to work on this because the DNA is too degraded. So using this, and it was the sequencer, and I was like, yeah. She's like, using this, we got SNPs, even though it was too degraded for stirs. And I was like, oh man, so close. So close. And so for weeks, anytime I'd be anywhere near headquarters, they'd be like, hey Meredith, you got any stirs? I tried. But I think that was my favorite anecdote where I was just like, oh, it was close.
That's funny. Was there anything that they like absolutely nailed? I mean, they listened to your advice on most of it.
He did, yeah. They got all of it right. They realized that, one base change, like a single nucleotide polymorphism, I told them that's a tiny piece. And if DNA is super graded, you can often still get those, but STRs can be really long pieces that are like 300 bases long. And so sometimes you lose those, and they got all that right. You know, they got like the kind of hair color and eye color stuff right. They got a lot of stuff right. I mean, it actually turned out pretty well. It was just that first time. I gotta be honest, like I didn't sit and watch all the entire episodes. I did, stuff to do.
Hey, I don't blame you. And it's something you work in every day, and watching a dramatized version of it is it could be challenging. There's been a number of shows, it feels like, in the recent 4 or 5 years that have been about podcasters investigating crimes, and that
Ooh, I
always
bet
makes
that's hard
me
to watch.
it always makes me chuckle. Another show about a podcaster. There's also a book about a podcaster, investigating crimes, solving crimes. And I think that too, to your point, they make it look really cool. And of course, there are quote unquote cool aspects of what I do as an investigative journalist and looking into these stories. There's also a lot of incredibly challenging and heartbreaking moments. And there's some really unsexy moments where I record in my walk-in closet with all of my sweaters around me. And, I have a dog who is a Velcro pet, but she's snoring. And so I have to ditch that record. There's just a lot. You can have a number one podcast and still record in your closet. And it's not always, sitting in a broadcast quality studio somewhere in LA. Yeah, so
Yeah,
I
I'm
go.
I'm always struck by how good looking we all are when they portray us on TV. I mean, we're some fine looking folks.
Yeah, between solving these crimes and doing every single step of the investigative process, you
In
have this
5-inch
love
heels.
interest. Yeah, you have this love interest and the romantic tryst, but that's the beauty of television. I don't think I think we both know what it is, we accept it for what it is, which is entertainment on the surface. But as a professional, I'm sure those little things that just kind of irk at you, that the stirs that which is what we call oysters here in my house, stirs.
Oh, all right then. Well, you know,
um,
could be a tasty snack, could be a DNA profile. You don't know.
yeah, there you go. Let's see. What are there any other pieces of the process, the forensic DNA process, or really anything that's done in a lab as it relates to criminal investigation that's been glossed over in pieces of work that you've seen, pieces of entertainment that you've seen?
I think it's kind of this like we have access to everything, you know? Oh, we can do that. I know a guy that can tell different kinds of dirt apart, you know? Or which kind of, but not exactly. I think things are obviously a lot more conclusive because they're not going to make an episode where we're like, "Oh, I guess we'll never know." That's not, that's not an end. They leave out a lot of the ambiguities. Um, I think that they tend to generalize that they have, any crime lab you're watching would have access to all of these things. Why wouldn't you do that? Well, they don't all have access to everything,
Right.
but budgets vary a lot, staffing varies a lot. Um, uh, yeah, we already talked about how it's not the same person doing all of those things. I, I actually met um the coroner for uh for Las Vegas, the actual coroner, many many years ago. He's probably not coroner anymore. I don't know, but I met the actual coroner, real dude, and he goes, "Yeah, I tried to watch the show about Vegas." He goes, and then the coroner pulled up in like a brand new bright yellow Hummer, and he was like, "Yeah, I'm out." He's like, "I don't get paid that much." that's that's not how it is here.
Right.
This ain't that. Yeah, we're not all running around in Louis Vuitton heels and custom tailored Armani suits and driving extremely nice vehicles kind of
I
a
mean,
thing. I
it would
think
be
it's
nice if that was the reality, but it's not. But that doesn't
also
make what
we turn the lights on in the lab like the overhead lights, it's not just like the blue mood lighting from under the
Mm,
counters.
mhm.
Turn the lights on usually when we're working.
Some nice
Helps
overhead
helps a
fluorescence
lot.
is nice. Yeah.
Hmm.
Yeah, let's go back to the discussion of these dramatized TV shows simplifying the investigative process as one person doing every phase of it. And
Yeah.
we've talked about previously how that's not the reality. Um, but can you kind of break down like who does what once something gets to the lab? Would one person follow one piece of evidence from start to finish, or does it get split up? Is it designed that way to protect the investigation, or let's kind of talk about who does what
Yeah,
differently
so that's a
compared
really
to
good
TV.
question, and the answer is it's going to depend on the institution. So some places work what they call cradle to grave, where one person will look at the actual evidence, they'll um, take the cuttings, do the DNA extraction, quantify it, get the STR profiles, then it goes to their supervisor for tech review, but they would handle everything else, and then they'll testify if it goes to court. Other places do batch processing. And so a lot of times this happens in bigger labs where, you have a whole bunch of people doing their evidence techs. They look at the evidence, take all the pictures, make the cuttings. The cuttings go to the next step. And then there'll be people who run DNA extraction robots and extract, extract, extract. And then it'll go to the next group and they'll do all the quantification. And then it'll go to the next group and they'll do the STR profiles in big batches. If you have a high case volume, that batch processing is more efficient. Um, some people like to work case entirely from cradle to grave and really take ownership of it. I think that can help if you're like, oh well, I remember what I saw back here. If you're the same person, and you're like, maybe I should go back. You don't have to ask somebody else. If you wanted
Hmm.
to go back to a piece of evidence, you kind of remember what was there, you took the notes. But the batch processing is going to be faster. And so if you're running a big lab with a lot of samples, batch processing is going to get more samples done more quickly. So it's going to kind of depend. There are pros and cons to both. Uh, the more you automate things, generally that's a good thing because, you know, robots do what their programming tells them to do. They don't kind of your a robot mind doesn't really drift off. I mean, it might have a malfunction, but I might be like, what am I gonna have for lunch? So it's good to take the not hard to do, but easy to mess up work and put it on a robot. And then leave the hard to do work for a human with a thinking brain. But yeah, that's gonna depend. Sometimes they'll work it all the way through. Sometimes they'll do it in batches, and sometimes they'll have rush cases, and then it'll normally be batch processed, but they'll do a rush case all together at one to make it go through faster. So
So I think
just
this
kind of
might
depends on the lab.
my next question might be a little off topic, and maybe I'm still thinking back to our previous session, talking about the timeline of DNA from collection to result. But um, in speaking of who does what, the interpretation phase, to me, when I hear interpretation, I hear matter of opinion,
Mm.
informed opinion. Um, would there be multiple people, evaluating results and coming up with their own interpretation? What happens when there's discrepancy? And I know this isn't really CSI effect, but it is speaking to who does what in the process. So I'm curious about that phase.
Yeah, that's a great question, and yeah, we probably should have talked about that in the last one. Me.
Now you're getting it now, so you got to
So
watch the whole series.
yeah, now you gotta watch. You get never skip part two. So the analyst that generates the DNA profile will do their own interpretation, standalone. Then they send it off. It has to be tech reviewed by somebody else. That's that's an FBI quality assurance standard rule. That's why the fewest number of analysts you can have in a lab is 2, because somebody does it and then the other person reviews it, or vice versa. If the person doing the tech review disagrees with your initial analysis, then they will often take it to the technical leader and have the tech lead review it, if that's a different person than who's doing tech review. And then they can all discuss it and say, well, this is why I thought this was, this was my conclusion versus that. And then they will go through their interpretation guidelines and make sure that that whatever interpretation follows those guidelines. Sometimes stuff just gets weird. And sometimes DNA profiles are hard to read. So it's not ambiguous in that nobody knows what's happening. It's more just making sure that whatever conclusion you're going to put out in the world follows your interpretation guidelines, so that you can avoid your own internal bias and anybody else's. And sometimes, unfortunately, that means you have to say this is inconclusive, because it's so messy that we don't have a guideline that will allow us to pick this apart. Or there's just not enough there. Like, you could see it, but it's below our threshold, and so we can't have confidence in that in terms of the way that we review our data. So it's kind of unfortunate, but yeah, somebody does the initial analysis, and then it's always tech reviewed by somebody else. So there are kind of checks and balances for that. And yeah, you don't really see that either. They're like, here it is, and they walk it straight to the courtroom.
Right,
And then interrogate
exactly.
the guy while he's on the stand.
Yeah, someone is cast as the scientist and they get the answer and they are the
Yep.
source of truth, as the show reaches its dramatic conclusion. So yeah, I think obviously, long story short, it's not just one person making this happen. And the
Nope.
CSI effect, quote unquote, has sort of colored, public perception that is how it works or how it can work, but that is certainly not the norm, if at all, a reality anywhere.
Yep.
Let's
Yeah. And I think another thing, with the CSI effect, it's hard to show a lot of DNA technology, because a lot of it is, a clear substance dissolved in a clear liquid in a clear tube. Like, it's not very exciting, like we're talking about with our previous series. And so they try to show things that you can see. I mean, we struggled with that. They would send people to the lab when I was in the R&D lab at UNT. They would send people and say, hey, I'm here to take a picture for the newspaper. What can you show me? And you're like, well, like, none of the DNA stuff is cool looking. Not really. We'd make like liquid nitrogen smoke for them and things like that didn't have anything to do with what we were doing. And so I think if you're producing television, you're going to look for stuff that's cooler looking. And so you're going to look for visuals. You could line up 2 fired bullets and look at at the marks on them from the rifling in the gun, that kind of thing. That looks cool on TV. You could show 2 hairs and say, oh, these were from the same person, which by just microscopic analysis is not really a statement that you can make. You could say these are very similar and they're not, but that doesn't necessarily mean one way or another that they definitively came from the same person or from 2 different people. I think that there's a lot of that stuff. There were a lot of things that were done kind of before DNA came on the scene that later turned out to be a little bit overstated. And, there were people wrongfully convicted based on those things. Also, I would say, like, eyewitness accounts, and this may be off topic, but eyewitness accounts are not great. Like, really not great.
Mm-hmm.
I was taking a forensic biology class, probably 200 people in the auditorium. And this is how long ago it was. The professor had a slide projector malfunction. Yes. because I'm old. And so he was like, "Oh, man, it won't hold on." And so he calls classroom services. And we're all just talking amongst ourselves. And the person from classroom services comes down, pokes around on the slide projector, declares it fixed, and then leaves. My professor said, "All right, everybody take out a piece of paper and write down a description of the person that just fixed the slide projector."
Mm.
200 people in a room with no other entertainment of any kind going on, sitting there, ostensibly watching this guy fix the slide projector. Guy. This person from the descriptions was short, tall, skinny, heavy, with a hat, brown hair, blonde hair, male. Most of the time, people said it was a dude. Some people said it was a chick. And we didn't have any, we weren't fighting for our lives. We were just hanging out. and the eyewitness collective description was not good.
No.
Clothing was all over the place. T-shirt, different, wrong, all different colors, all different, wrong. Finally brought the person back in. Yeah, it was a fairly average-looking uh, guy, but we never could have nailed that dude as a class of people studying forensics.
No, was that an intentional slide malfunction? I'm so fascinated by this. It was all a matter of proving a point.
100% staged
Wow.
to
That you
show us that.
Yeah. Yeah, exactly. Yeah, that's another aspect of the CSI effect of, if we're stepping out of the lab, of course, the dramatized version is only showing us pieces of the investigate. Like, there's very rarely a red herring thrown into a dramatized TV show, is what I've seen. There's scarcely a piece of evidence that isn't related because that would take time away from the precious 30-minute window.
Yeah,
And there often is, there's a lot of it is up to interpretation or understanding. If something is there at the scene, is it connected? I just covered a cold case, actually a case from 1941, one of the oldest I've ever gone back to. It's unlikely to get any investigative attention moving forward, but the only piece of physical evidence collected at the scene was a red handkerchief. And this red handkerchief caused me so much just strife thinking about it, because like, well, what could that mean? Anything? Is it connected to the scene? Was it just happened to be there? And one of the frustrating things about this case being so old, all the records and files were destroyed according to retention you know schedules.
yeah,
And so I never got to learn anything more about this red piece of this red handkerchief. But that's unlikely something that would have made it into a dramatized version of this story, because there was no closed end to it, and that
yeah.
often
I mean, they're not
happens.
going to spend 10 minutes on somebody analyzing this piece of evidence and at the end go, "Yeah, well, what about the red handkerchief?" And they're like, "Oh, yeah, no, that wasn't part of this at
No, it
all."
didn't mean anything. Right. Exactly.
I
um
mean, cigarette butts, especially when people smoked more. Um, okay, there was a cigarette butt on the street. Okay, well, yeah, there were 50 of them.
right.
Was
Yeah.
that from then? Is it from that person? Was it just random? Is it just people because people litter there? Like, I mean,
Mhm.
that's rough.
Yeah, and that's
How do you
another
know?
part of the investigation where, the people on the scene are determining, is this evidence, you know, relevant? Do we collect it? Do we send it to the lab? Do we use those lab resources to run something that we're not sure about? It's all these little this nuance to an investigation that doesn't make it into a 30-minute or 60-minute drama. I think it's
Yeah,
interesting though, where the crime content sphere has really grown in the last 10 years is the area where I work in, the true crime, investigative journalism based podcasts. And we've seen more and more documentaries that are including firsthand interviews and talking to the investigators, talking to family and witnesses and things like that. So I think and maybe this is my own bias because I don't seek out dramas anymore, but I think we're moving in a direction where some of this, the misconceptions, hopefully, are being dispelled by more true, real, investigative content out there. Have you seen that as well?
I think so, and I think, yeah, I think the existence of podcast as a medium has really helped that because it takes, if I'm making a television show, like I'm not going to do that on my own. It takes a studio and all this and writers and people, executives and everybody's involved. But podcast as a medium, or podcasts as a medium, I think allows people like you to select their own content and say, this is what I want to know about,
Mhm.
and to share that more directly with other human beings. And people are very interested in what really happened. And I think it's wonderful. I think that shows kind of the the breadth of the kind of the real human experience, other than just, these things that can be wrapped up with a tidy bow, to show like, hey, some of these things we still don't know. We may never know. People are still talking about the Jack the Ripper case.
Right.
People still want to know. I
Hmm.
mean, I think that's, yeah, I do think that it's kind of moving that way. And I think that, you know, now all of these kids have the internet. I sound old, but I mean, there was no mechanism to do that. Not really. Um, when I was in undergrad, at least, I think I do think it's kind of moving that way. And we see some of it. I don't know. I'm still trying to decide how I feel about CrimeCon, and things like that. I haven't been. Maybe I should go before I decide. Um, but it's obviously people want to know real things that really happen and not just, made up dramas.
Right. Right. Meredith, do you have any advice for someone their interest has been sparked by either a drama or a documentary or a podcast, and they really want to get into the forensic side of things? They're interested of, maybe pursuing it as a career, making a career change. What's your advice for someone who wants to get into this field?
That is a great question. I would say start with your aptitudes. Like you're interested. You're interested in criminal justice as a broader topic. Start with your aptitudes. Um, you know, I will meet somebody and 12 seconds later, while I'm still talking to them, I don't know their name anymore. I am I, I will meet somebody, see their face, talk to them, not see them for 2 months, see their face again, and think maybe that person looks familiar, but I'm not gonna be like, oh yeah, that was Bob that I met at this conference this last time. I don't have a great memory for names and faces. Investigators do. I mean, really good. It's not one of my core competencies. There are people who are great at like physics and math and accident reconstruction. Really relies on that. There are two cars coming. This one's going this fast. What really happened there. Um, if you're, if you did really well, if you really like chemistry, and that was something that you were good at, toxicology. I mean, all the drug chemistry testing for, arson agents, that kind of stuff like that. The kind of chemistry side of it. If biology is something that you were good at, or that you learn easily, like that was me. Um, going to the DNA side of it made absolute sense. I was into molecular biology before I was into forensics. And so this was me applying kind of my abilities to what there is. Used to, you couldn't get a forensic anthropology job because there are not that many of them, and those dudes never retire. I think they just die like there, like in the office. I don't know. I mean, I guess sometimes eventually they do, but they stay for a long time.
Right.
And so you couldn't get a job, but now that's starting to turn over. And so if like anatomy and physiology was, you were good at that, or you were doing pre-med stuff that um the pathology part was very interesting to you. Those jobs, that kind of a is a different angle on all of that. Like what happens to, how do you look at a deceased person and find out what happened to them? Autopsies or autopsy techs or pathologists or something like that. And so for me, I pick up stuff related to DNA and molecular biology very easily because I find it super interesting. I don't have to make myself learn it. It just like soaks in, you know. I mean, obviously I put a lot of time into it, but um if I tried to go get a bioinformatics degree, uh like you'd have to hold a gun on me. It's just not my core competency. I understand it as a field from a little bit farther back, but I'm not going to be somebody that's going to be innovating new techniques in bioinformatics and looking at big data. So when you hear, um, people talk about genetic genealogy and looking at all this big data, that kind of stuff like that, that's not one of my core competencies. People that are good at that big data stuff have applied that, like from you know different career paths. I know a very well-known lady who was in kind of nuclear physics and just was used to working with big data and applied that to genealogy because it's just a lot of data points and looking for patterns and kind of connecting the dots. And she's really good at that and she's made a career of it. So, take a look at your core competencies. What do you like doing? What do you want to do every day? Because I definitely have seen students who were interested in criminal justice in general, but they got into their first lab class and realized that transferring small amounts of clear liquids from tube to tube and very precisely was going to be a lot of their job, and they didn't like that. Like that wasn't good for them. That wasn't a good fit. And so, I would say take your core competencies. Everybody's got kind of aptitudes. Um and then those things are the going to be the easiest for you to develop into a skill and into a career. And so there's no single path, many paths to the top of the mountain. There's no single way to get there, and there's no single way to help out. Some people are better at the social aspect. Um, and so, like, different kinds of criminal justice degrees and that kind of thing. Some people, and being investigators, that kind of stuff. Um, some people have real good fine motor coordination. I can't catch a set of keys if you throw them at me to save my life, but I can
Same.
move 2 microliters. I can move a half a microliter. Perfect. No problem. I don't shake, nothing, and an aptitude for understanding DNA, like that's good for lab work. Bioinformatics is definitely up and coming for forensics. And as we start sequencing things more, there's going to be more data that needs deconvoluting. And so if you're good at that more kind of statistical analysis and those other things, that might be that kind of mathematical aptitude, but you also understand the biology, that might be a good way for you to go. Just take the things that come more easily to you, and then look and see where to apply that, where it makes sense, because there's a whole bunch of work to be done. There's no shortage of work for sure.
Yeah.
Um, so I don't know, that's very vague, but a lot of, you don't have to start out with a forensics degree. You could start out with a degree in physics or chemistry or biology or criminal justice or something else, and go from there. And even if you didn't have that, you can still, there are a lot of technician jobs that have very different requirements. You could kind of try it out and work as a tech for a while and see if that suited you. And if you wanted to get more education to do the next thing or to do something else, you could do that too. So
And I think on, my side of
it's
things,
something for everybody.
if anyone watching is interested in this work but is not a Meredith, is not a numbers or chemistry or physics or any side of the, of that, the science piece of it. There's also opportunity on, journalism degree, or even without a
Yeah.
journalism degree, a content creator can responsibly tell these stories and help to combat that CSI effect and demystify the processes that are actually getting these cases solved, an active current case all the way to a 30, 40-year-old cold case. There's so much work to be, to your point, there's so much work to be done, from the actual investigator standpoint, the forensic scientist standpoint, but also from the storyteller perspective, really amplifying the cases that aren't getting attention, becoming a platform for families to honor the legacy of their loved ones who have been lost to violent crime, unexplained disappearance. There's such a spectrum, a universe of opportunity to contribute to this space if it's something that your interest was sparked by watching a 30-minute crime drama. And I think that's the
Yeah.
big point to drive home, is that if your interest is sparked and you want to make a difference, there's a lot of opportunity to do that, no matter what angle you're
And
coming from.
I completely forgot about all the computing science stuff. Um,
Everything. Yeah, there's everything.
I mean, yeah, triangulating cell phones, seeing where devices have been, and following the money because that usually helps. Forensic accounting, all that kind of stuff. There are a million disciplines.
And it's all needed, as we've seen.
Yep,
It all plays a role in getting answers for these families. Well,
absolutely.
this has been another I love that word, illuminating conversation with you, Meredith. It's been really cool to hear your perspectives, your anecdotes from your time on set. I had no idea we had a Hollywood lady in our mix. So thank you for sharing your time with us. I'm sure you need to go sign autographs or something. Uh,
Yeah. No, I never even got paid for that. So pretty sure not famous.
Well, thank you again. This has been another incredible series in our DNA deep dive, and I'm looking forward to what comes next.